And our second speaker is Anandhi. Anandhi Iyappan. She got her microbiology, her bachelor in microbiology at the University of Madras, and also make a master in bioinformatics, and also in life science informatics at the University Bonn. She had made her PhD in applied life science by Fraunhofer, and she's also since May 21 member of GHGA and metadata workstream. And this is a pleasure for me having you today. And I hand over to you. Thank you very much.
So this part of the presentation will focus more on communities and data portals. So in the first part, we obviously looked at the importance of fairness and what one needs to do to ensure that the data is fair. But now we will basically be exploring why do we really need these community data portals and what good use do they bring to research. So in general, biomedical research projects deal with a lot of data management requirements from multiple sources like funding agencies, guidelines, publishers, policies, and they also have their own best practices. And of course, the user needs are also very different for each project. So obviously, to have any kind of a portal or a support system that helps in managing research data and digital artifacts from individual researchers or groups of researchers are definitely a great way to ensure that research can be more, the research data can be made more reusable and interoperable. So having said this, let's first explore the existing communities that are investing in open research or providing infrastructures for storing research data. Okay. Okay. The first initiative that I would like to talk about is Elixir. So Elixir is a European national bioinformatic infrastructure that enables researchers to assess and analyze life science data. It underpins the management and safeguarding of data that is generated through publicly funded research. The main purpose of Elixir is basically to help or ensure that that the data that is being produced can be found, analyzed, shared, and also we have a lot of group of experts who are also there who form communities to discuss task forces and individual domains of interest, which helps the community to also expand and also to have deeper discussions. This particular Elixir community is distributed well across Europe, the research infrastructure for life science. There are about 23 nodes spread across 250 organizations. And since Elixir has come into existence, there are also many trustable domain-specific repositories that have come have come through Elixir or have been collaborating with Elixir. And you can see the list of other communities that are already part of it and are kind of in a big pool where research has been constantly exchanged, and Elixir has played a great role in kind of supporting so many data repositories. And obviously, the last part about Elixir is supporting also the data repositories that has to be fair. And that's why they also make use of standards, services, fair services. They also take a lot of effort in curation and verification of the data. So the community is enriched and well equipped to support diverse research projects. The next community that I would like to talk about is the Research Data Alliance, which was founded in the year 2013. An international organization that has a certain focus on infrastructure and community development, mainly built for enabling good data sharing and exchange. Research Data Alliance also has various focus groups or cross-group community groups where the discussions those communities are always having, they revolve around various topics like reproducibility, data preservation, best practices, data citation, metadata, and data type registries. So it gives people, or it connects people with different research backgrounds, but dealing with similar problems. So that's about Research Data Alliance. The next community that I would like to talk about is the fair sharing community, which I'm quite sure most of us on this call might be very familiar with, especially if you're familiar with fair data. So a fair sharing community basically guides and users and helps them to discover, select, and use the resources with a lot of confidence. That was the main reason why fair sharing came into place. And they basically are nothing but a curated and informative educational research on various standards like data and metadata, interoperable, interrelated, which has certain interrelations to databases, and also have a lot of policies in place. They also develop a lot of policies and standards within the community. And the open standards, obviously, they also have various they come up with various guidelines, recommendations, terminology, and also exchange formats, helping again the research community. They have developed so far about 1,500 or 1,700 standards, about 2,000 databases, and also they have come up with 266 policies, which can be easily adapted and reused by various diverse research communities. And the next one, which is probably a more, I would say, a recent addition to what we call as research community system, is the Global Alliance for Genomics and Health. It's GA4GH, it's shortly called, is a nonprofit organization that builds a foundation to broad and also ensures responsible data sharing, especially when it comes to genomics data. GA4GH aims to accelerate the progress in genomic research by developing standards and also framing policies. And they have certain core objectives for this, which revolves around data sharing, interoperability, privacy, security, ethical and legal considerations, and innovation. I haven't mentioned metadata, which is also one of their work streams. And they also have a certain life cycle that they kind of support that kind of very beautifully bridges the connection that exists between research and the clinician world and user communities. And that's why those core objectives are also covered in the form of user groups, which makes the community broader and active. And they all support in fostering human genomics and health related topics. So now that we have seen a lot about community portals, we also need to understand, okay, there are communities, but why do we really need, or what is the need for fair data portals at the first place? As we all know, and since we are all involved in research, we obviously know that there's always a growing demand. There's a lot of data that is being produced, but all the data that is being produced, we do not know the quality or the quantity is obviously high, but we are always unsure about the quality of the data that is being produced. And therefore, fair data portals take extra care to ensure that the data that is being produced can be well stored, managed, and also have the possibility to share them. And all of this can only be done in a reliable and a trustworthy manner. And this is what differentiates normal data portals and fair data portals. And most of the fair data portals have a certain archiving functionary in their data portal, an ability to retrieve data, ability to analyze data. And they're always supported by help desk systems, which always address user requirements, and also a strong ethical and legal community, which focuses on all the ethical aspects of the data and the administrative aspects involved with it. So, having said about fair data portals again, before we also venture into the different types of data portals, we should also first understand what are data portals, right? What are open data portals? Data portals in general are platforms which are designed to store, publish, and share data from different applications and users. And they ensure that they provide the infrastructure in such a way that even researchers who do not have a lot of computational background find it very easy to submit their data, find it very easy to search information, and also ensure that the portal is designed in a more transparent manner, which helps the users as well as the developers who contribute to research. And data portals generally follow not all of these points that have been mentioned in the slide, but they always ensure that the data that they are storing are discoverable, measurable, accessible, have metadata that is publicly available. They always promote the use of standards in storing or capturing or linking their data. Documentation, good documentation, user documentation for users to have to easily understand the structure and submission process that is involved, and also various tools like an analysis wing, which focuses on how to visualize and analyze the data that is being produced. So now let's look at some of the existing data portals since genomic research or biomedical research. NCBI Geo is probably one of the oldest, but also one of the earliest data portal that existed. The Gene Expression Omnibus is a particular database repository where one could have know a lot of information about high throughput gene expression data and chips, arrays, and also microarray based experimental data. It's a public archive, and also it's a resource mainly for gene expression data. So in a platform like GEO or NCBI GEO, it is possible to search in their search bar. And with that, one would also be able to filter various domains that one wants to capture, or how they want to customize the data sets that they want to search for. For example, in this use case, a hepatocellular carcinoma was being searched for, and that can also be customized further by various categories like what kind of organism we want to study, what should be the study type, who's the author, and so on. And then it will help you result, or it will give you a filtered list of those experiments based on your choices. Apart from that, it also provides a place to do some analysis. GO2R is also one a plugin that they have in their data portal, through which one could also further analyze the data sets that they have downloaded and compared with their own local data if they have not yet submitted to GEO. So this provides the user to also directly compare their data with the existing data in the data portal, but also helps them to retrieve data from the archive that related to gene expression data. I already mentioned about visualization. Whatever data has been analyzed, it can also be visualized. So it gives researchers an instant result of the data that they want to analyze. And of course, it's also possible to download data set and do extensive analysis. The next data resource that I would like to talk about is about Genomic Data Commons. The Genomic Data Commons, shortly called as GDC, is also a platform that was developed for supporting cancer genomic studies. So even in this platform, it allows users to search, analyze, and download data, and it is also very specific to human cancer data sets. So this also focuses on a lot of cohort-based studies, and it also has a cohort builder that is inbuilt in this data portal, which helps the users to also filter data sets based on their requirements, like clinical information, biospecimen information. And it is also possible for users to customize cohorts and make their own cohorts for their research purposes. The third and the last data portal that we would be looking at is the CBio data portal, which is a platform also for cancer genomic data sets. They have a very explanatory as well as an interactive, very good visualization for those data sets. Here, they allow data sets. They also have a query interface through which can be customized. And they also allow researchers to interactively explore the information about genetics, and also networks, 3D protein structure, and so on. And they also combine literature as well as data coming from other data portals. And they also have information coming from clinical data, omic data. So it gives a researcher a multi-omic integrated information, which helps the users to also learn a bit more about existing data sets or the data sets that are there, and to gather more information related to the data sets that they might be in need of. Having said about, or having looked at the data portals and the communities, now let's also explore if there are ways to assess fairness in data portals. If at all they exist, what are those? So before we go into that, we first have to understand why all of fairness matters in general. For that, we have to understand the importance of data quality, right? So if we have poor data, what implications will it have? So obviously, as of now, we always have an increasing amount of research that happens, and all of this research are either done collaboratively or individually. And if those data are not properly documented, not properly maintained, not properly stored, obviously what we are giving into these data portals, that's what we will also retrieve out. So garbage in, that goes into the data portal. If it's not good quality data, when a user comes and searches for information, he's also going to get poor quality data. So what will this impact, or what kind of impact will this create? It will obviously create inaccuracy. It will cause a lot of redundancy. It will lead to incompleteness. And obviously, this whole exercise is expensive. See, as and when we provide or produce more and more research data, if they're not properly maintained, then we have to keep reinventing the wheel, and it is going to hinder us from progressing in doing better research. So it's very important that we take enough measure to have good quality data sets. So it can actually enhance research, but that does not hinder the research in any way. So one way of assessing quality of data is through FAIR metrics, or we can call them as indicators. So when we are talking about data portals, it's important to actually first understand what kind of indicators do we want to create. There are some customized indicators available, like for example, to have a metric ID, a metric name, to which principle does this metric apply, what is exactly being measured, why should we measure it, and how do we measure it. This is just a very simplified list, but this can obviously be expanded based on the study purpose, based on the project, and based on the use case that is being developed. The RDA, the Research Data Alliance, they also have a metric ranking based on choosing the metrics by marking them as whether they are useful, important, essential, or non-essential. The FAIR community has also developed various metrics like the FAIR metric, which is nothing but a toolkit that basically enables the community to kind of see whether the data that is being produced actually passes that metric or not. They also have a list of indicators in them, and when run through a data, it will actually show that metric is high, low, or medium. And this can be done for both automated as well as manually generated. The FAIR metric works for manual as well as automated assessments of data sets. The second one is the RDA, which is the Research Data Alliance FAIR Data Maturity Model. They also have developed a FAIR Evaluator, which helps engaging the data management services and it basically supports and helps the developers who develop tools to build stronger FAIR services. The third one is the FAIR Checker, which is also something that was developed by the FAIR community, which assesses the FAIR principles and empowers the data provider to enhance the research data quality. So what they basically do here is that they check that the data, whatever data is being submitted to them, they check for the quality of the metadata. They actually check whether the metadata that is being captured has stronger ontologies and customized vocabularies that have been used. Based on that, it will give a good ranking, and it will mark whether that data quality is good, bad, or medium, or good, bad, or unusable. So these indicators obviously are very useful because it helps the user to choose high quality information, and also for the data portal to have a stronger standard in accepting that the data from diverse communities or from researchers per se. So since we're talking about data quality, there are also certain metrics revolved around data quality. So these principles identify basically what needs to be there, right? But they don't need to tell what is really necessary and what is really sufficient. So evaluation of these fairness basically reflects to the extent of which the data resource addresses the FAIR principles in general. So first of all, why should FAIR metric even be in place? Because it helps in better discovery and reuse. It helps in better interoperability of data. And they are usually measured, or these quality metrics consist of these 5 indicators, if we can call them. One is the completeness, second is correctness, conformity, consistency, and coherence. This obviously can also be expanded, but this is the most widely accepted quality metric as far as FAIR principles are concerned. So when I speak about the metric, that metric can also be measured through scores. That scoring can also be a certain indicator. So whatever metrics are measured here, they can also be quantitated by some kind of a key performance indicator. And how can that be done is by kind of, whenever a data set comes in, all of these metrics can be checked for, right? Like, for example, when we talk about completeness, we can always check if all the metadata that is being submitted are actually present and captured. So this basically says that this data can be findable, interoperable, and reusable. Already that strikes 3 out of 4 measures of FAIR. Then it can be checked for correctness, that is, all the data that is being captured have been captured correctly, or if there are any mistakes. So this supports a lot of reusability of data. Then conformity actually speaks more about the if all the data that is being captured actually confined to certain standard, each property or each class that is being captured is confined to a certain standard, and this obviously enhances interoperability. The next one is consistency, whether all the data that is being calculated are actually consistent, which means is it making any sense, the whole data set making any sense. This supports a bit more of the reusability. And coherence, basically to ensure that all the data elements are actually coherent across all the different data sets, the data domains that are being captured. In many cases, we have, you might have seen in various portals, there is exhaustive spreadsheets, and sometimes data is linked between different spreadsheets. And if it is not coherent, then it's not going to make any sense. So that coherence is also very important. And this basically supports all the F, A, I, and R of FAIR. So such metrics, obviously, like I said, already transforms the data from just being a data to a high quality data. So I just want to also show you a small example of how this can be done. So when you want to perform a completeness check of a data, now let's just look at a data set. We have two data sets here, ABC1 and ABC2. And here, the first one, we have captured all the values for each property. So the first ABC1 has age, sex, tumor subtype, tumor location, and previous treatment. All of this has certain value. If you look at the second data set, it has information about age, it has information about tumor subtype, it has information about the tumor location. But it does not capture information about sex or the previous treatment. This is one example. And then we have the correctness. The correctness here is what? So the first data set also has information of under sex. It has wrongly captured this. Instead of capturing the sex, it has captured the ethnicity or the orientation of that particular individual who was part of the data set. That's wrongly captured, so that doesn't qualify for correctness. And in the second data set, you do see that the age and the tumor location are not properly captured. In case of age, it's not captured as an integer, but it's captured as a decimal, which is not the right way to represent the correctness of the data. So when we want to assess this through a score, we can obviously score for the first example. All the values for ABC1 has been captured correctly. So we can give a completeness score of 100% because 5 out of 5 has been right. And in case of correctness, 4 out of 5 is correct. So we can say it is 80% good quality, 80%, the data's quality is 80%. In the second example, we have seen that 2 out of 5 features are, 3 out of, I'm sorry, there's a mistake, I think. 3 out of 5 features are correct and 2 are wrong. So that is 75%. And in case of correctness, 3 out of 5 are correct. So it is 75% again. So like this, we can create a certain percentage ratio to measure the quality of data sets. This gives, even as us, as people who are data providers or data portal developers, on how we should capture the data, and even for a data user, for him or her to be able to choose the high quality data or the data set that they want based on a certain score that is being assigned for each data set. Apart from this, there are also other ways to do quality control. So one other way is something like we can also do something called as a syntactic check, where it is helpful to actually do to check whether the semantics that are being captured and the syntax that have been used in relations are actually captured properly or not. And this obviously can also favor efficient data reuse and interoperability. So the example that you see here is an example from biovalidator that is being developed by the EBI, where they actually do a check they have a checklist of what they call as a schema. And that checklist can be used to validate against the data that is coming in. And these schemas can be, they can automatically check and see if the schema matches, or if there's a mismatch, it will always indicate to the data developer of the data portal. So all of this enhances or supports data portals to get good quality information without having to do any manual checks, because that's going to be a very laborious process. So all of these checks will actually help automation of the quality of data that comes into the data portal. So one other example that I found very interesting was how data portals also focuses on how they can support data submission. So ENA is one particular data set which has a certain interface for creating and reviewing submissions. And since this also deals with human data, we just wanted to show how they basically do or how they have designed the portal for data to be submitted. So they have, of course, they have a controlled access submission because it involves sensitive human information or human data. And this database consists of various functionalities like the submission and update of the XML metadata objects, the spreadsheet-based submission for the different classes that they capture. So this is how their portal basically looks. They have, what you see in the first is the schema. On the second, that you can see the type of study that you want to register. And the third one, you can actually have talk about the kind of sample that you have. The fourth one are the type of reads that have been that were part of the experiment, and the fifth is the analysis that were being performed over the data set, or the features involved about the analysis. So if you want to submit a data set to them, you obviously have to create a login, a secured access, and of course a password. So only if you give the right details, and you also have to be affiliated to an institution, then you will be able to log into the app and you will be able to submit your data. Am I still audible? Because I just saw that there's, okay. So they actually come up with a supported metadata checklist, which means they have also an exhaustive list of that one they have captured as a minimum submission guideline for different kinds of submission. They have, like, for human associated, they also have separated them across different body organ parts, like anatomical parts, human gut, human oral, human skin, and this is also very exhaustive. So this also customizes submission based on the type of human data that you want to submit. This list that you see here is just a part of it. Of course, it's much more exhaustive than what you see here. And they also have a lot of customized spreadsheets for the type of experiment that you want to submit to them. So their spreadsheet looks like this. They have, under experiment, you can capture the different features, and they also mention whether those features are mandatory or optional, so which also helps the user to decide the type of information they want to provide. The same is with the sample. I'm sorry. Yeah, the same is with the sample. And so like this, their spreadsheet is well documented and well distributed and well described, which also supports the users to submit the data in a much more easier fashion. And since it's also customizable for different studies and different type of experiments, the user need not spend a lot of time to fill in all the information, but actually can customize and submit the information that he has. So this is one portal that I personally found very interesting, and I wanted to share it with all of you. Caro mentioned that I will be talking about the difference between the real world data versus the cohort data as well. So it's just some that we basically know that we will be dealing with two types of data sets in any case. So we just also have to understand what they are. So in case of the real world data sets, it is actually very important again, because it enables the researchers to not just think about their own traditional like control trials, but actually to go beyond the data that is being gathered through that way. And here it can actually be collected from any number of cohorts or any kind of population or subgroups. But the problem with a real world data set always remains that they follow their own protocol. They have their own schema that they use to capture the information. And also it becomes very difficult to integrate anything that they create with the existing systems that actually exists. So this is probably one of the disadvantages or a limitation of real world data. But in case of cohort studies, it has a certain very clearly structured, it has a very clear hypothesis defined in the very beginning. And therefore everything about that study is thought through well in advance. And also the data related to these variables can actually be measured because the study is so well thought through. Everything that will be captured will be done in a very systematic manner. But again, in something as so nicely structured, they also come with certain limitations, like there is no room for any kind of randomization. And this also impacts the kind of relationships, the cause and effect relationships, and it is very difficult to determine them. And also the relationship between variables must be obviously be stated as associations. It cannot be done beyond that. So it kind of lacks a certain, how do you say, it is not as flexible as the real world data. But again, both of them come with their own advantages and disadvantages. So that's something, again, we wanted to talk about here. Now that we have looked at the data portals and the communities that support, I think right now what is also very important for us to talk about is the project that we work on GHGA and why the model that we develop actually matters. So in GHGA, because we have also done this whole exercise of trying to compare, Caro did mention about this, right? So you don't have to always reinvent anything. If there are things that already exist, we can always make good use of it. So that's kind of the similar exercise we followed in developing the metadata model. So in this particular model, we also ensured that we have standardized concepts, and we base our model based on concepts which already exist, but also we ensured that we did not create anything or we did not customize anything on our own. So it is called a standardized system that we have adapted for this. And this is also obviously modeled using certain framework that also describes the semantics and also has a certain structure to this. We also use a framework that supports semantics and standards. This is the schema overview of the metadata model. So our model consists of these classes, like the study, the data set, individual, sample, experiment, and research data. So this model will obviously be, it will evolve with time, but this is basically the core framework or the core backbone of the metadata model that we have. And if you want to know more about this, we also have done a webinar previously where we gave a detailed explanation about the model. We went into each aspect of the schema and how we capture them. So if you would like to learn more about it, please do visit our channel and learn more about the data model. Why GHGA matters at all at the first place is that this model is also well aligned with standards, not just nationally, but also internationally. As I told you in the very beginning of the metadata model part, the GHGA part, that we did not really start or develop a model from scratch. So for us, the goal and the vision was to be aligned with the European Genome Archive. So our structure and the skeleton of our model closely resembles our EGA. And we're also in touch with international community like GA4GH, and we are also trying to ensure that we adapt similar standards. So we are well aligned with the current state of the art communities and the efforts that are being taken in those communities. So we also looked at this in Caro's presentation a bit more on how to develop a metadata model. So we also have our own constraints, but at the same time, we also want to be fair. So we have created the model with a balanced design, which supports both data reuse as well as takes enough measures to protect the data that comes into our data portal. And we have two different use cases that we are primarily focusing on. So our model can also be customized for the modalities that come to us. Right now, our focus is cancer genomics and rare diseases, but in the future, we will also be able to accommodate different other disease communities as well. And last but not the least is the qualitative metadata submission with varying degrees of freedom for the submitters, because this is what we also insist always, that we do not want to make the submitters' life very difficult. We want to support them with as much customization as possible, but at the same time, we also want to ensure that the quality of the metadata that we receive is good and can be reused by other researchers as well. So to summarize, the community-based initiatives in general play a very, very important role in establishing FAIR concepts across the scientific community. And with FAIR data portals, it greatly enhances the visibility. It improves the reproducibility and also adds a lot of confidence and reliability on the research that is being, the kind of research that we do. Any kind of metrics or indicators are extremely important and very, very essential to have a check on the kind of quality of data that comes into a certain data portal. Of course, the degree of freedom will obviously vary depending on the sensitivity of the data that you're dealing with. But such metrics are great indicators, and it's always important to ensure that we can adapt at least some of them, if not all. And obviously, very, very important point is to ensure that the design of metadata model always balances the quality of the submission, but at the same time also promotes the openness and also any kind of advancement in scientific research. Because in order to create a perfect model, we might have to do a lot of compromises. So the balance is extremely important to maintain, that we think about enabling better research, but at the same time also ensure that we are not compromising on anything. So that's also very important. With this, I would like to say thank you to all of you, and also for staying with us for so long. We also have some additional resources that you can also dig in if you want to know more about what we are currently doing. We have already published a white paper, and the model that I showed you in today's presentation is also part of this white paper. But right now, we have already launched another version of the metadata model. You can also access that in our GitHub, from our GitHub. And also we have a user documentation where you can actually read a bit more about how we have developed the model, and all the details about the recently developed model are also present there. With this, I would like to thank all of you once again, and I'm happy to take any questions if you have any.